Okay, I like the sense of community here. People have a lot to talk with others. Thanks for coming, and it's been a while since I saw you last. I hope you had a good time with the presentations of Lior, and before that, Ron. I think it's a good time to clap and say thanks for that. And so today we'll talk about rates, and I have to do it at a quick rate because there's lots of good things. Before that, I'll say that the rate of progression of the course is pretty fast in the sense that there's actually only 2 more lectures that I'll be giving, and then I'm passing the baton to you for the final project. So maybe a few words on how that's going to work. We'll send you everything in details, etc., but just so you start to get a perspective. Each one of you is asked to suggest a question that they want to do a small calculation about, like a 5-minute thing that they'll be presenting in front of class where they'll be, getting the opportunity. I know it's a bit intimidating, but you'll see it works okay. We'll also send you a few filmed examples from last year and previous years where you're supposed to take a question that either is just you're curious about, we'll give you examples, or something that relates to your research, which is even better if it works that way. And then you're just presenting sort of like, what's the question, maybe something about the motivation, do a small calculation, and then look at the result and think about the implications. That's the idea. And the way we want to do it is like we really want to respect the time of all the other people listening, so we want you to prepare well for it. So the idea is to find somebody who would serve as your mentor from the class that would be, like they do in reality shows, would help you go through it, and you'll be preparing with them. A week before, you need to send us, what feedback the mentor gave you on how to make it better. And with that, you'll come here, you'll be presenting. The other people will be listening and then sending you feedback. And that could help you improve your final project, which is not only what you present in front of class, but also like a short write-up, a vignette of like 2, 3, 4 pages, kind of like what you read in the book that you'll be handing in. And again, writing the motivation, the calculation, the implications. The idea is kind of clear. Probably not completely, but we'll give you more information. We just wanted you to start preparing mentally to that. And we'll send you the plan in terms of when would that happen. So it would happen in the same time slots of the course. So Mondays between 11 and 1. It would be like something like 5 such occasions. And we'll allocate each one of you to that, and we'll be sending it to you. If somebody has any constraints that they can't, or this or that, let us know through the website email if you have any constraints. And that's about it. We probably would be running in terms of the semester, because it's like 5 meetings or so of your presentations, we'll be running into the exam period. So if you have exam schedules for Mondays 11 to 1, and that's a problem, also let us know, and we'll try to take that into account. Good, I forgot something important. Okay, so let's jump into the scientific part. Let's start with something that's very much on my mind, and that's the rate of climate change. And I want to see if there's a way to try and derive like a quantitative, get a quantitative feeling to how fast is climate change happening. And with that I mean, well, anybody remember something about what is the expectation on the rate of climate change? Let's say by the end of the century. Okay, so we had 2, 3, and 4, which sounds like not a big deal, but it's like a huge difference whether we'll stay below 2 or we'll reach 4. But that's kind of like the range that we're talking about, which could have like really catastrophic implications in terms of reaching tipping points of the Earth system, but that's not the subject of the lecture here. What I want to talk about here is to try and get like an intuitive sense of what does that mean by converting that rate of change in time of say 2, 3, 4 degrees by the end of the century to something that is more intuitive to us, and that's distance. So the question is, how do I convert between a temporal change, and the spatial change, a change in space, which is something we're usually quite, in terms of rates, it's quite intuitive to us. That's the challenge. And I suggest to approach it in the following way. Let's think about our blue or green planet, Earth. We have the equator. And let's say I want to see, so the temperature of all this is going to change by, let's say, 2, 3 degrees. Actually, if you look, by the way, when you're saying, say, 2 degrees, it means that on land, it would change roughly 4 degrees. Because when talking about 2 degrees, it's the average globally, and that's both land and water. And actually, the oceans warm more slowly because they have more inertia and all sorts of other reasons, they're deep, etc. So they usually warm up more slowly. The land warms up more quickly. So if on average it's 2 degrees, it's actually about 4 degrees on land. Okay, so let's say I know that I'm going to get a change in temperature of roughly 4 degrees Celsius. And that's during, let's say, 100 years. I'm interested to think, what does that mean about how quickly, let's say I'm standing, let's say somewhere here. And let's say I was, right, we know that like here the temperature is relatively high, and here the temperature is relatively cold, right? There's some gradient between the equator and the poles. Okay, so if everything is warming up, it's effectively, if I'm looking and saying I had one temperature here, it means that a year later, it's as if it's getting warmer, like the temperature, I'm becoming closer to the equator. You see what I mean? Yeah. And I'm interested to do this conversion to say, how quickly do I see the temperature changing? Any suggestions how to approach that? Exactly. So what would you say? What's the difference here in temperature?
40 degrees.
Okay, I like it. So the change in temperature between the pole and the equator is roughly 40 degrees Celsius. Okay, what else do I need to know? I need to know something about the distance, right? Okay, so what is the distance between this and that?
So the radius is like 6,000.
So the radius is about 6,000.
6,000.
I think you already know all the answers in the course are usually 10. So 10,000 square kilometers. Hey, not square, 10,000 kilometers. Okay, that's roughly the circumference, or like 1/4 of the circumference of Earth. I think to a pretty good approximation, it's about right. Good. So now I have this, I can do like a small analysis and say, okay, if I'm looking at the gradient, which is the change in the, like the length per change in temperature, 10,000 kilometers, 40 degrees Celsius, that's about 250 kilometers per degree. What does that mean? It means that if the Earth was completely flat, and still we have this difference in temperature, every 250 kilometers that I'll be walking towards the pole, it would be 1 degree warmer. Or 250 kilometers towards the equator, it would be 1 degree warmer. Okay, which means that, like, across Israel, which is about 500 kilometers from side to side, at the longest end, it would be like 2 degrees difference. Okay, so, it passes some sort of sanity check. Good. By the way, you see that I've used 2 significant digits, which usually I recommend not to do. I would usually write this 200 or 300 or something like that, but you'll see for a second why I'm still motivated by that. And that is because now I want to go and connect that to what I'm seeing here. So what do I see here? I see here that I have a change of 4 degrees in 100 years. Okay, so this is the spatial gradient in space. I can also calculate the gradient in time. And there I have, in 100 years, no, I want to do it the other way around. I have 4 degrees. 4 degrees in 100 years. So that means 0.04 degree per year. I can now derive from these two things the thing that I was interested in, which is the velocity of climate change, which is roughly what?
10 kilometers.
So the way to think how to combine them, you could look at it directly from the units. What are the units of speed? It's kilometers per year, right? So I have here kilometers per degree, and here I had degree per year. So if I just multiply them, I would get what I wanted. So 250 kilometers per degree times 0.04 degree per year, and I'm getting 4 times 250 is 1,000, but I have it 2 decimal points to here, so I'm getting about 10 kilometers per year. We can also do it in like just, put in some, that's one way to think about every year. The line of equal temperature moves by 10 kilometers. If you're standing in place, it's as if you got 10 kilometers closer to the equator. Or if you want to stay, if you're a tree and you want to stay in the same habitat in terms of temperature, etc., you should walk towards the pole 10 kilometers every year, except for the fact that trees could not walk. But you have to disperse by 10 kilometers, which is a pretty fast thing, given that your lifetime is usually many years. So you need to, your progeny, if they want to be born at the same place, they have to be born like 100 kilometers north or something like that. 10 kilometers per year. It's also, just to make it, it's good to play with different units, about like 1 kilometer per month. Right? Or how many hours do we have in a month? So we have 30, 24, so about 1,000. So that means it's about 1 meter per hour. I don't know where's north here. North, I think, is there. So by the end of the lecture today, if I want to stay in the same temperature because of climate change, I need to move 2 meters to here. That's roughly how much, climate change is progressively moving. Yeah, so it's kind of, at least for me, it's kind of mind-boggling to think about it also in this more dynamic manner, which I think, is less abstract. Of, by the end of the century, something would happen. You could see, every year, it's moving forward. Okay, any comments, thoughts? Yeah.
It feels like the calculation doesn't take into consideration the fact that it's not a linear expansion, but it's more of an exponential growth of climate change, from what I understand.
Okay, so there's a few things to be said here. One, I think that is a very good point that, this could convey, okay, so we'll need to just move 10 kilometers every year. It's actually very tough to move humanity by 10 kilometers every year because we have our houses in a specific place and it's hard to move it by 10 kilometers or 100 kilometers after a decade. Not to talk about, water levels and all of those things. But even beyond that, this thing that I mentioned before about tipping points of the Earth system, this could be catastrophic in all sorts of ways. Things could change from this very simple kind of like linear effect. Okay, so that's a good point, and thanks for raising it. There's a different issue, and that's the thing, we're mentioning about exponential or not. Just as it's something that comes up very often when people are talking about like exponential process versus linear processes. I want to clarify something. If you take an exponential curve, it's usually denoted this way, right? And you look at a time window that is not very large, what you get is something that it's exponential, but it's actually not very different from linear. And that means that, all these things that often people like to talk about, exponential, it depends on what's the duration. Over a given, if you have enough of time, it's a big difference. But actually, in many cases, it doesn't matter so much, and actually, you might even not be able to tell so much. And so I would say for climate change, it's bad enough, even as linear, and if it goes up by 2 degrees by the end of the century, global average. And whether it's exponential or not, for me, it's like actually nitty-gritty details. I don't know. It might be accelerating or decelerating. It's hard to say. It's going up in that direction with that gradient, and we should be worried. Yeah.
But due to exponential is on the distance between the equator and the pole. I mean, it is a significant distance. It's 10,000 kilometers if you assume that the temperature goes linearly from the north to the...
Okay, so there's two. Okay, now that's also important in communication. There could be different interpretations. What I was interpreting, what Erez was saying, is that the change in temperature of the Earth as a result of climate change with time is being claimed to be exponential. Okay. By the way, I don't think it's like, I'm not sure whether it's true or not. In the sense also that exponential, usually you need to have a few doublings. Like there's no way to do a few doublings. Like humanity would not be around. In a few doublings, also, you do it in Celsius, you do it in Kelvin. What does it really mean? Anyway, it's accelerating, and that's a big problem. Okay, you're referring now to how the gradient in temperature across the globe, how does it go here? Okay, so we could look at it. I would challenge us. I don't think it's exponential. Also, we could look how linear, not linear. It's pretty it's monotonic, and I don't think it's I don't think there's a crazy dynamics there. But we could look into that, and maybe there'll be some factor of, and then what would happen? Let's say you're right in there. So instead of 1, it would be 2, or 0.5, which is kind of like the thing I don't care so much, Okay, we're good? At least in that respect. You might remember that last time that we were together, we gave you like a question to test our intuition, also about rates, and that was about what is faster. Transcription or translation. Where here we were talking about whether it's the process of transcription for RNA polymerase or the process of translation through the ribosome. Okay, and we were talking about several options. Anyway, we could, let's do a quick vote. Who's voting for transcription being faster? About half. Who's voting for translation being faster? Another half. Who's voting for roughly the same? Few people. Who's voting for it depends? Okay, we got rid of this option. Okay, so most of you are wrong, which is great. So let's try to put numbers around it. So just again, we're talking about, let's say, DNA, we're already at RNA. No, you have, I could start and do the schematic, let's say. This is the DNA, and then we have the RNA polymerase making here the RNA. And then on the RNA, we have the ribosomes jumping in. And they'll be going in this direction. And from there, and as a result, you start to have a protein. How is my schematic? Clear enough, right?
Accurate.
Okay, so now we're asking about the rate at which this goes, it doesn't matter, in this direction. This is the rate of transcription versus the rate that we have here, which is the rate of, it's to translation by the ribosome. Okay, anybody knows anything about those rates?
Few kilobytes per minute by polymerase.
Okay, so saying for the RNA polymerase,
I remember about 40 base pairs per second.
so I have 40 bases per second. That's about right, which is what you said when you convert it. So let's say few tens to 100 bases per second. Okay, well, I'm again using the notation of this being a few. We had 40, so 40 sits somewhere here. It could be 60, it could be 30, it could be 80, somewhere in this range. Okay, what about the rate of the ribosome? Anybody carries something in their head? Okay, so you might see it in this, like we had this, I think we might have sent it to you, the key numbers in cell biology, which is just like a few numbers that we think are worthwhile remembering, because from them you can infer, instead of remembering 1,000 numbers, you need to remember like 10 numbers or 20 numbers, and with them you can do lots of stuff. So the rate of the ribosomal translation is actually quite useful by itself, and that's somewhere between 10 and few tens of amino acids per second. Now you might ask me, why are the rates this? And I'll tell you, I don't know. It's the empirical fact that you can do the measurement and see that these are the rates. The understanding of why did evolution reach those values, or why biophysical constraints limit those values, that's a really good question, and I think very worthwhile investigating. There's some papers about it, but I think there's more to be said about it. Also dictates many things like the ability to, how fast can you divide, and other things that you could explore. But I don't have a great answer, but it's the sort of thing also that I like in the sense that it raises new questions from looking at the numbers. But I can infer something about going back to this question. What can I infer? What? That it's the same. Why the same? Because this seems faster than this. Right, so you can see that between this and this, we have like a few fold difference of this being faster. But there's a small catch here, that this is bases and this is amino acid. And for every amino acid, you need 3 bases to be transcribed, right, in the genetic code. So interestingly, the ratio between this rate and this rate is also this ratio, meaning that effectively they're going at roughly the same rate. Okay, any implications for that come to mind? That what? That it's simultaneous, exactly. So if you look at what happens here, it's basically the rate at which this, think about it dynamically, which I'm not so good at now making it a schematic, but when this goes and this becomes longer in terms of message, the guys, so there's several let's say in bacteria where it could happen concurrently, you'll have them moving at roughly the way this length, this also moves. You'll have more of them now accumulating here. But the rate at which they're moving is roughly the rate at which they're getting the message, and it could also all happen simultaneously. And indeed, if you have like all sorts of images, you see that it's like beads on a string accumulating there, and like it continues from roughly where it stands here, and you create these nice, it's called Christmas trees. It's really, really cool when you have like good electron microscopy of those things. Yep. Yarden?
Also, infer something about the amounts of the proteins. Like if the translation, for example, was much slower, then you needed to have a lot of ribosomes to be able to translate all the RNA that they get.
Okay, so you can start doing things to say, you could play the game of say, okay, what would happen if translation was much slower, if translation was much faster? It's actually easy to start with translation is much faster. If it was much faster, what would happen?
There would be bottleneck.
What?
Bottleneck. There would be a bottleneck.
Yeah, there could be a bottleneck, or basically, this like, it would reach here, it would get stuck, which is kind of like a bottleneck. And if it's much slower, then okay, you'll have a gap here, and you'll need maybe, you'll need more ribosomes maybe in order to be able to do it. So you can start and do the analysis, the accounting of what would happen in each case. Yes?
But what about the mRNA contains untranslated regions?
Excellent. Excellent. So what I've shown you is the picture in bacteria, like the simple. Now we're going to the advanced version. Okay, advanced version is in eukaryotes, and now actually it doesn't happen concurrently. in the sense that you have this happening in the nucleus and then being exported, and there's something happening outside. So then it's not at the same time. And you also have untranslated regions like the introns and other things that now expand this. And that's true. So then the picture becomes more complicated there. But the rates are not very different. What's the main difference in the rates, by the way? The thing that's affecting the rate is actually temperature. Let's say if you're looking at yeast versus bacteria, the 30, usually they grow at like 30 degrees, and this grow at 25 degrees, this grow at 37 degrees. For every 10 degrees, you have a factor of about 2 in terms of the rates. That's the main thing that's affecting. Good, so now, for example, you can say, okay, how long does it take to make a protein? How do I derive that?
Where do you start?
300 amino acids.
So I have about 300 amino acids, good, for an average protein.
About a minute.
Okay, and then I'm doing, let's say, 10 amino acids per second. So I'm getting about 30 seconds or 1 minute in order to make a protein just based on doing the, say, transcription translation if they happen concurrently. And again, if you have also introns and other things, you could have initiation, you can have the export, you can have all sorts of things jumping on top of it, but the hardcore process is about a minute. Does that mean that I would always be able to respond in 1 minute? Let's say I'm having some message to a cell, and then it tells it to produce some protein. Would the response be in 1 minute? Why not?
You need to deliver the message.
Okay, so I need to diffuse in order to deliver the message. Okay, so it's interesting to calculate that. Maybe we'll try and do that. Is there anything else at play here? What? The RNA needs to find the ribosome, or the ribosome needs to find the RNA. That's also, so they need to diffuse until, whoops, it sits on it. Good, that's another thing which is worth calculating.
The protein is diffused to the place of target.
Okay, so we have, good, so we have a lot of things that need to diffuse. We'll get to there.
You also need more than one protein to make the signal be effective. It's usually kind of an amplification process in signal transduction.
Right, so I'm interested in this for a second. I actually won't have time to dwell into it. If somebody wants, there's a great course by Uri Alon in the book on Introduction to Systems Biology dealing with it, but just a few things to be said about it. If I want to have a response, there's a question, what would be the response time? And there, in order to talk about it, we don't need climate change for that. If I'm looking as a function of time, and I'm interested in the level of protein, and let's say I had somewhere here that I had, something that triggered. Okay, so there's the diffusion and all the other things that will take a while, and then I'm starting to produce, and then we said, okay, another minute, and then I'm starting to have the first protein being produced, right? So I have here a delay, which is on the order of like a minute plus the diffusion time. But then, just like was pointed out, this is not immediately what you'll be responding to. Now, this needs to accumulate. And then let's say this is the steady state level. There's a threshold at which, at some point, in which I'll start to respond. Usually you take it like, say, half of the maximum or something like that. And only this would dictate what's my response time. And the question how long this takes, this could be way more than a minute because you need to make many copies. And then there's really cool stuff about how to derive those things that actually turns out to be in relation to the degradation time of the protein or the mRNA, which is not very intuitive, but lovely. And I won't have time to get into it. If you want, in some previous lectures I got into it, but today I have other stuff I want to show you. And the best way to learn about is, for example, looking at Uri Alon's lectures or book. Good, how are we doing with time? We're in good time. Okay. So let's see what we talked about the finals, about the rate of climate change, which machine is faster, the time to protein, the response time, and you mentioned diffusion. So let's talk about diffusion. And my question is, how long does it take to diffuse across a cell? In the spirit of the question that that was relevant. We could leave that question there, and we ask, how long to diffuse across a bacterial cell? Meaning, if this is my cell, and now let's say I have some signal coming from the outside, and then it tells something, I know, the transporter or the whatever is sitting here, and now I have change in, could be phosphorylation of a protein or something else, and then this thing needs to move, let's say, from here until the other side or until the nucleus or whatever you have there. Okay, if bacteria, no nucleus. And let's give some options. Is it less than a microsecond? Or is it between microsecond and millisecond? Or between a millisecond and a second? Or between a second and a minute? Or between a minute and an hour? Question is clear? Okay, let's test our intuition. Who's voting for less than a microsecond? 1, 2. Who's voting for between microsecond and millisecond? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Who's voting between millisecond and a second? 2, 4, 5, 6, another 10. Between a second and a minute? 1, 2, 3, 4. Who's between a minute and an hour? 1, 2. Okay, a very pluralistic class. We have quite a range of answers. Before we dwell into that, let's ask the same question about mammalian cell. Okay, so same drill, but just somewhat bigger cell. Microsecond, microsecond to millisecond. I have a few. Millisecond to a second. Half second to a minute. A few more, and a minute to an hour. Few. Okay, so the question is, how do I find it out?
We're talking about passive diffusion, right? Not
We're talking about passive diffusion in contrast to what?
in contrast to an active search process that involves also diffusion.
Yeah, passive diffusion.
It also depends what diffuses. Is it a protein?
What?
Is it an ion?
Yeah, so it depends on what would be diffusing, although I'm not sure how much it would change the timescales. We could think about it. So let's say we're talking about a protein, just to make it fun. So what we need to do is to derive, to have an equation. For that, and for that there's something called the diffusion equation, which I can throw at you. But just for the fun of it, let's see if we could derive it together. Okay, so let's take an analogy. Okay, because we're going to do something for diffusion happens in this thing called random walk. Who heard the term before? Okay, most people. But then the question is, what are the implications of random walk? So let's try to say, I'm interested to know in a given number of steps or a given duration in time, how far do I get? I'm looking for an equation that would connect those two.
Are we assuming that it already tests the membrane?
Yeah, let's say, yeah, the membrane already, yeah, you pass the membrane, or maybe you connect on one side of the membrane, and then from the other side, some message, some magic happens that transduces the message, and then you did, say, some phosphorylation, some other message, and then the question is how fast it would diffuse from there onward. Okay, so the question of how far do you get, we could take like a real-life analogy, and that is, let's say I'm the particle that needs to diffuse, and I'm doing random walk. Random walk means that I walk this way, and then this way, and that way. And now let's say I need to get from here by 1, 2, 3, 4, 5, 6 steps. I'm reaching the cell boundary. And the question is, I guess here we have about 30 or 40 students. The question if I was taking, so with 6 steps I could get there. The question is, with a random walk of, let's say, 30 or 40 steps, like the number of students here, would I be reaching it or not? Or how far would I reach?
Majority of the times you would stay in your place.
So, you know what, let's do it.
Normal diffusion, sometimes you'll be there, sometimes you'll be there.
Yeah, you're right. So sometimes I'll be there, sometimes I'll be there, sometimes I'll stay in place. So let's try and do a simulation of that. Okay? So each one try to think if they're born on an odd or an even day of the month. Okay? And now let's see. Even. So even, let's say it's 1 step to here. Odd is here. Yeah? Odd? We said odd is this way, yeah. Odd. Odd, wow.
Odd.
Odd.
Even. Even? Even? Very strange class. Odd, we said this way.
Very odd.
Odd. Even.
Odd. Also, it works on special muscles, yeah. Even. Yeah. Odd. Odd. Odd is this way here. Odd.
Odd. We're getting close. Even. Even. Even.
Even.
Even.
Odd.
Whew.
Odd.
Odd, we said this way. Even. Odd. And our, okay. So you see, actually, it's not very far from what you said. I didn't stay in place. I got somewhere. I feel my muscles. And we saw that I got some distance. And the question is, can we calculate what's the expected distance that I got to? It seems like I didn't get to 6 steps in the same direction, which is interesting. I didn't know to predict exactly how it would work. But actually, I did have a prediction, because I know what's going to come out in the equation. Okay? So let's try and see if we could derive it together. Okay. So this is me. Okay, in the first time, it's like coin flip or anything. I could either go a direction, let's call it A to here, or direction here, minus A. Right? So this is a time 0. This is going to be time 1. I'd like to look at time 2, time 3, etc. So in time 1, I could be at either A or minus A. What are the probabilities? 50-50. So 1/2 to be here and minus 1/2 to be here. Okay. What about time 2? At time 2, I can already be at, I can either go here or here. So I'll be 1/4 of the time I'll be at 2A. 1/2 I'll be at 0. And 1/4 I'll be at 2A. Okay, this is 2 minus 2A, 2A. Okay, so far so good. Okay, I have a 0 chance of being here and here, right? Because it's whatever. Okay, let's do one extra step. So from here I could go to 3A. What is the chance for that?
An 8.
1/8. Or I can go back to A. What is the chance to be here?
A quarter.
So the chance coming from here to here is 1/8. But then there's also a chance of coming from here to here, which is 1/2 times 1/2, so 1/4. 1/4 plus 1/8, it's actually 3/8. Okay, that's fun. Believe me that it's already also true in this direction, right? It could also be here. So my chance of being at 3A minus 3A is also 1/8, and to be at minus A is 3/8. So far, so good? Okay, and you see it's symmetric, and it sums to 1. 1 plus 3 plus 3 plus 1 is 8, divided by 8. So all is good. Okay, so now what can I ask here in order to get an equation? So I'm interested what would be my location, right? So location is x. I can ask what's my average location. This symbol is like taking an average. So what's my average location? It's minus 1/2 times, 1/2 times minus A plus 1/2 times A, which is 0. And I could do it to the other things, 1/4 times minus 2A plus 1/4 times 2A plus being at 0 is again half. So you see, whatever I do, I'm getting 0, which is the point that was raised before. What's your name?
Eli.
So what Eli was saying, that actually I'm, on average, I'll stay in place. Okay, because I'm going to the left and right, same probability. So I didn't get very far. After all this derivation, I got 0. So not a great equation. So what can I do?
The mean of the square.
Yeah, so I can think about the square or the deviation, the variance, things like that. Okay, might sound familiar from physics, statistics, all sorts of things like that. So what you're curious to say, okay, let's do the same thing, but just at x squared. And the nice thing with x squared, it's always positive. So it's not very probable that I'll be left with 0 because I'm summing things that are necessarily 0 or positive. Okay, let's try and do that really quickly. So I have, if I was doing the same thing here, it's 1/2 times minus A, but now squared, plus 1/2 A squared. So that's A squared, 1/2, so it's A squared. Okay, that's my expectation for the distance squared. Okay, let's do the next one. 1/4 times minus 2A squared plus 1/4 times 2A squared. So that's 4A squared, 4A squared. 1/4, that's A squared. So this is 2A squared. Okay, A squared, 2A squared. Okay, let's do last one. 1/8 times minus 3A squared plus 3/8 minus 8 squared. And I have the same thing from the other side. What do I get here? That's 9A squared divided by 8 plus 3, it's 12. And then I have another 12, so I have 24 divided by 8 squared, which is 3/8 squared. And I want to continue this until I reach n steps. So what's going to be my equation? I can repeat this, but I think you're becoming tired. So it seems like it's 1, 2, 3. I think you can believe me, it's very probable I'll get NA squared. And of course, we could do the math to do it properly, but in the spirit of this course, we're not taking all sorts of, we can calculate the Gaussians and do some derivations and stuff like that. But you can see that kind of like street fighting mathematics or equation derivation, you can see what happens in diffusion and what I'm getting. And I'm getting that the distance squared is equal to the number of steps times A squared.
This is one-dimensional diffusion, though.
You're right. But let's not confuse ourselves with one dimension versus a few dimensions, but it's not very different. Okay, let's finalize how do we get an equation? So what is this number of steps n? Can I write it differently? N is really a number of steps. It's like the time that I'll be taking divided by total time divided by the time step. Okay, like it's how many, like how long does it take me to take a step times, you know, this ratio is the number of steps. Okay, so now what do I get here? I have that the final distance is equal to a squared divided by delta t times tau. This is often denoted as r squared, the distance squared that I'll be taking. And what about this? What dictates how much a squared per unit time I'll be passing? Let's say now I'm going back to, let's say I'm a protein inside the cell. So there's all sorts of things that are going to happen here, like the size of the thing that is moving. Is it an ion or protein or something else? The temperature, what?
The density of the material.
Yeah, the density inside or the viscosity, all sorts of these names of things, how things move inside other things, hydrodynamics, etc. So I don't care about all these things. Actually, the physicists don't want to get into it so much as well. When you're writing the equation, you just take all of this and you call it d for diffusion coefficient. And you end up with the equation of r squared equals d tau. And this is the diffusion equation. And now we could take this and implement it in order to try and derive the answer to these questions. What do we need to know?
D and tau.
What?
D and tau.
D and tau. Okay, so let's talk about D in a second, but tau obviously we need to know because tau is what we're looking for. So maybe we want to change the equation, and instead of r squared being equal to d and tau, we're interested in knowing tau. So what is tau? Not so complicated. I just need to divide here. It's r squared divided by d. Because that's what we were asking there. You see the connection, what we're looking for? So what do we need to know? We need to know r and we need to know d. Do you know what r is? Let's say for the bacteria, 1 micrometer. So what do you want to know? D. How much is D?
How did you assume that it's 1 micrometer? I didn't follow that.
Because I want to diffuse across the cell of across the bacterium.
The radius or rate?
No, R here is the size. Okay, it was the Rx squared, so it's a size squared. Okay, it's a bit confusing because R can sometimes denote rate and sometimes denote distance. I'm talking about like radius or length.
Okay, so
Maybe it would have been better to call it L squared or whatever. Okay, so you know what about D? How much is D? You already know the values in the course. It's about 10. But you know, it's kind of a coincidence, but this is the case. D is about the diffusion constant, let's say for a protein inside the cell, is about 10. But then what are the units? The units are micrometers squared divided by second. That's another useful number to know, like the diffusion coefficient of things within cells. How do I know this is the units and that it makes sense?
I do not understand.
So just 1 second. So if, for example, I'm taking D, I'm plugging it here, I'm multiplying it by seconds, and then I'll get units of micrometer squared, which is, again, it's the size that I'm looking for. So that fits well. We'll also look here. This is the units of this. So I have size of this, let's say micrometer squared divided by micrometer squared, and the second would go up, which is exactly what I'm looking for. So in terms of units, I'm okay. You would also see this stated sometimes not in these units, but let's say centimeters squared per second. So and we could do the unit conversion, but I really like this, both because it's like small, nice numbers like 10, but also because micrometer squared is the relevant time length scale. So you could work in what they call MKS units, like meters or centimeters, something like that. But actually, there's something nicer in working in units. You just need to be consistent, which are relevant for the microscopic world. So micrometer is a good unit. Okay, yeah?
There was someone that mentioned it before. You said that, let's not get into it, but I just wanted to know, is D the same for 2-dimensional and 3-dimensional space, or is it different?
Yeah, so the D is the same, but in the final equation, the way it's usually defined D, you actually have also a factor of 2 here for 1 dimension, or a factor of 4 for 2 dimension, or a factor of 6 for 3 dimensions, which is not very intuitive. Maybe it is somewhat intuitive, because if you think about how far I'll get, the variance, it's kind of like you can think as independently, I'm moving in this direction, and the random walk is also happening in this direction. And if I was able to jump, you could also think that. And then all of this is like, every dimension adds another factor and to the variance, and then it becomes 2, 4, 6. I don't know if that's very helpful. For the spirit of this course, we want to know kind of like the order of magnitude. You don't need to care so much, because a factor of 2 here or there would not change the result significantly, because I'm asking you, is it microsecond, a millisecond or millisecond there, or second to a minute? Okay, so we'll take a break, but for the break, I can just...
Just ask about D. Is
Yeah.
that something you get from experiment, D10?
Yeah, so how do you know this? Yes, of course. That's. I think you can't derive it from basic principles. Maybe you can tell stories after the fact, but basically it's being measured, and you could use like GFP and look at a very good microscope and see how it's moving about, and from that you derive it. Okay, well, there's other methods, not just GFP. Okay, so thanks for that question. So, okay, I'll just give you one challenge if you want to try and do while you're in recess. And that, given what I gave you here, try to revisit your answer here. Okay? Good. Let's take a break. Okay, welcome back. Did anybody have an opportunity to try and update their estimate, or try and came up with something?
At this point, just calculate the numbers in the formula.
Yeah, so, okay, we could just plug it into the formula, right? Okay, so let's do tau bacteria. It will be roughly what?
1 to 10.
So r squared divided by d. So it's 1 micrometer squared divided by 10 micrometer squared per second.
0.1 second.
So it's about 0.1 second. So,
20 milliseconds per second.
yeah, so we know that we're here. Okay? You see, by the way, that I'm not being very particular about whether it's 10 milliseconds or 100 milliseconds or something like that. And that relates to many things. There's this issue of whether it's 1 dimension, 2 dimensions, 3 dimensions. There's a mention, somebody was telling me about the fact when I was doing the derivation, it's not completely fair, because after you enter into the cell, you have the cell envelope somewhere. So if you take a left, you get stuck. So you don't doing like the complete random walk. You're doing a random walk where every time you hit the wall, you're stuck. So you can do the calculation, doesn't matter so much. But it's the style of thing that therefore I'm taking like a big range. Also, like this protein and that protein, do they have the same diffusion rate? Maybe it's not a protein, maybe it's a metabolite, and the diffusion rate might not be 10%, maybe it's 100 in those units. But all of that sits very comfortably in this big range of a factor of 1,000, so some number of milliseconds. And then we could bring us back. You remember the question we said, okay, when I was talking about the transcription, how long would it take until I'll have the response? And many people were raising all sorts of aspects related to the diffusion. Diffusion of the ribosome to this, of the signal to there, of this to that. Everything's negligible, right? Because if it's going to take you a minute to make this process, all those milliseconds, completely negligible, which is nice. It shows like this timescale separation between this process and that process, which I love. Okay. Let's do the same thing for the mammalian cell. Remember, we had the same question for the mammalian cell. So how much is that? Okay, so I need to know the size that I want to diffuse over. How much is that roughly? 100 micrometers? That's a big, it's true, some of our cells are 100 micrometers. Let's say the current, what? Few tens. Okay, good. So, few tens micrometers squared divided by what? Still 10, exactly. So the diffusion rate does not change. 10 micrometers squared per second. So how much is few tens squared? So few times few is 10. The 10 and 10 is 100. 100 times 10 is 1,000. I have 1,000 micrometers squared by 10 micrometers squared per second. So it's roughly 100 seconds. Okay, so about a minute. So it's sitting somewhere here. So it's in green. So somewhere between D and so like the beginning of E. Now there's something interesting here, right? The size, the difference in sizes is they're different, but not by a factor of 1,000. Anyway, so you're getting also a rule of thumb, so it would take you, say, a minute in order to do diffusion inside the mammalian cell. Good. Okay, so now there was mention of different types of whether the cell is 100 micron or 10 or a few tens of microns or something else. Let's talk about the specific cell type now. And these are axons. Anybody knows anything about axons?
It's not a cell, it's part of the cell.
Yeah, right. I'm talking about neurons and the diffusion across the axon.
Can I ask someone something about the mammalian cell?
Yeah.
Like, because it's so big, it's also divided like to little organelles. And
Right.
RNA not likely to move all the way to the membrane. It's probably just get out of the nucleus and already find the ribosome to translate.
Okay, so there's several things to unpack here. One, you're right, there's different organelles and compartments and things like that. And therefore, usually, maybe you wouldn't need to go the whole cell. Maybe you'll just need to go half of the cell. Okay, half of the cell, we could do the calculation. It's squared, so it's a quarter of the time. Okay, so instead of a minute, it might be 10 seconds or something like that. Then there's another issue, and that is the fact that on the way, you're going to bump into all sorts of things that have membranes, all sorts of stuff. And that's also true. That also would impact things of how the diffusion happens. The diffusion constant of 10 takes into account the fact that it's like this strange media inside the cell that bumps into many, many different things. But it might not take into account the fact that you have the ER that you can't penetrate into it, and that could bring another aspect. But you'll stay around seconds to minutes. Okay, it's not the milliseconds and it's not hours as a result of it.
Why did you say it's not a factor of 1,000?
Okay, so I was saying it does come out to be a factor of 1,000, but in terms of the size, I was moving from a bacterium that's 1 micron in length to, let's say, this mammalian cell that's a few tens of microns in length. So the factor in terms of distance that the thing had to diffuse was a factor of a few tens, not a factor of a few thousands or whatever. Okay, so now I want to do, the next step is, let's say instead of bacterial cell or mammalian cell, how long to diffuse across a 1 centimeter axon of a neuron. Okay, right, neurons, they have this they have these axons in order to shoot to the next neuron and do all sorts of calculations in our brain, our body. The length could vary. Some of them are, could be a meter long, from, I don't know, spinal whatever to somewhere in my leg or something like that. But let's take things more standard, let's say 1 centimeter in length, which also happens in the body lots of places. And now, well, you already know it wouldn't be a microsecond or a millisecond, so let's give you new options. So I think for the mammalian cell, we already had it. So let's have it between a minute and an hour and a day and a week and a month, or more than a month. Okay, the question is intuitively clear. I want to diffuse over across like 1 centimeter of an axon, which is part of the neuron. And you know, lots of things need to diffuse, right? It would be signals, proteins, metabolites, amino acids, you name it. And the question is, how long would it take? Okay, who's voting for a minute to an hour? 5 people. Who's voting for an hour to a day? 5 people as well. Day to a week. A few, also 5 people, but now the hands are quite low. Who's voting for a week to a month? 4 people. Who's voting for more than a month? Not even one. Tzaddik in Sodom. Okay, so these are the answers. So let's derive it, right? So I suggest you try to do it this time. So let's try to pair up with the person next to you on some side. And let's take 5 minutes to try and get an answer. Okay, I suggest we continue. Anybody wants to update the vote?
Yes, everybody.
Okay, so let's do the calculation. So the time to travel a 1 centimeter axon, what do I need to do? I need to plug in R. So what is R? 1 centimeter, but you know, the units I need to do the conversion, right?
10,000.
So I have 10,000 micrometer squared divided by the 10 micrometers squared per second. So that's 10 to the 4 squared 10 to the 8 micrometers squared divided by 10 micrometers squared per second. So I'm getting about 10 to the 7 seconds. Right? How much is 10 to the 7 seconds?
About 10 days.
It's a few months.
Few months.
I love to see your excitement at that. So the way I remember it, I remember this rule of thumb that is like pi 10 to the 7 seconds in a year. I don't know if you saw that in one of the. It's actually to like within like 1% mistake. There's nothing in pi there, but like you just do the calculation, it comes out to 3.1 something, say 10 to the 7 seconds per year. So 10 to the 7 is like 1/3, so few months. So we're definitely around this. And now the question is, what do we do? How does that work? Functionality-wise, yeah?
I think if I remember correctly, that's what would be if things just diffuse, but we have ion,
We also have
something that happens that
kinesins and
makes it
dyneins.
much faster.
Again, biology comes up with some magic.
Yeah, I can't remember exactly, but this is what would be if we didn't have one. See, myelin or something that insulates that it goes much faster.
Okay, so just to be clear, so we have the cell body here. We're talking about the axon that is here until it reaches whatever. This is the 1 centimeter we have to pass. Yeah, it's longer than this, what is sketched here. It's not in scale. And it's true this might be myelinated and stuff like that, but that's important for the propagation of the electric signals. But then this, what we arrive is what if you had like random diffusion of things from here to there. But there's this magic happening, and this magic is by having some other component of active mobility. It's called molecular motors. Anybody knows anything about molecular motors? Yeah, so there's different kinds of them, and they have these beautiful movies if you want to check it on YouTube, where like the myosin and kinesin, etc. They take some cargo, for example, amino acids or RNA or something like that, and then they start moving, carrying it like with these nice steps, and they got Nobel Prizes for whether asking the question, when it walks, does it walk this way or this way? You wouldn't believe how biophysicists get excited about this question and giving Nobel Prizes for that and everything. It's really fantastic. But whatever the method, the mechanism is, it works at a rate of about 1 micrometer per second. That's the velocity, roughly, of the molecular motor. So is that fast or slow in terms as speed goes? It's slow, right? So remember, when we calculated, to go a distance of a micron in the bacterial cell, it took us how long?
0.1 second.
Some number of milliseconds. So this is relatively, in comparison to that, that's like about 10 to 100-fold slower than the diffusion. So how does that help?
Not random, like consistently moving everything into.
Exactly. So if I want to reach that there, and in random walk, as you saw, it's kind of tiring. But if I go in a directional way, even if I go more slowly, but all the time consistently in the same direction, I would reach there significantly faster. Let's calculate by how much. But it wouldn't take me a month. And the wall is not here. Think the wall like at the other side of the building or something like that. Instead of taking a month, even though the steps are slow, because they're consistent, they don't have this randomness of going back, it will take how long? Anybody wants to do the calculation? 16 minutes? Okay, too many significant digits. So let's try and do it together. So the time using the molecular motor, so how did you derive it? It's the same length that we have to take, right? Of 1 centimeter. But now, instead of this thing with squared, etc., it's the usual equation, right? It's distance divided by speed. So 1 centimeter divided by 1 micrometer per second. 1 centimeter is about 10 to the 4 micrometer divided by 1 micrometer per second. So you're getting about 10 to the 4 seconds, which is roughly 1 hour. Okay, just say, so we see that like instead of a month, it takes me an hour. So on the one hand, I'm now walking 100 times more slowly, let's say on a micron scale. But because it's consistently in the right direction, I'm getting there like 1,000-fold faster. Which shows something about like, it's not very intuitive, but then, you can develop that intuition. There was a question or a comment, yeah.
Explain why you don't have to square that first step here?
Yeah. So this thing with the squaring was when we were talking about this mechanism, and we derived this equation, and that's for diffusion. Now we moved into the world of molecular motors, and the point that Yarden was making is that molecular motors, they don't do this random walk. They're always walking along the filament in one direction. And then it's back to normal stuff, velocity times time equal distance. And therefore I just need to, there's no square. Okay, so you can see the awesome usefulness of moving from diffusion into something directional in this manner. Okay, so we talked about axons and we talked about motors, and good. So before we wrap up, something that might be useful also for the exercise that you're getting this week. So just to be clear, if you want to get feedback on your exercises, you can go to the Moodle, and in the Moodle, in your private space or whatever it's called, you can find the feedback given to your exercises. Okay, and if anybody has other questions about it, come forward afterwards and talk with Sam or with Leo. So also in the spirit of rates, let's go back from the microscopic to the macroscopic and think again about the us and the world. And my question to you is, how many people are born in the world every year? Okay, we're done with transcription and translation. Just a small step and we have the whole world in terms of rate. So it's the kind of question that you'll be grappling with also in the exercise and beyond. But if you just think about this, how would you come up with an answer? Think for a second.
Some exponential function.
Okay, so we had something about these two options, or like some exponential thing, etc. Okay, but so good ideas, but let's say, I don't let you go out of the room without giving me a number. Okay, that's the spirit of what we're doing in the course. We're not just talking, in theory, we'll do some derivation of demographics with some exponential something. We just want to give some number and get out of the room.
So like the population is like 9 billion, say, and I would say like 1% people are born every year or something. So I will just give you a number and go.
Okay, and then I let you go. Okay, I think that's the kind of approach, and there's actually a nice way to approach these things, and that is the fact that if you want to estimate, it is like a relationship where the pool size is like the number of people in the world, is equal to rate times residence time. Okay, so for example, if it was an actual pool and you want to talk about the water there, if you know the rate at which you're putting water inside, and the average time that the water is in the pool before it gets out, this would be the relationship between these two things. Or in our context, if you have the number of the people in the world, is like the number of people being born per year, times what is the residence time? It's a number of years you're going to be around. Okay, so this relation holds. And now, if I give you, I believe now you can derive it and you'll get some more experience in doing such things in the exercise. But this is a very useful relationship. Why don't people use it more? Why didn't you use it? Yeah, it's very simplistic. And I think there's something, in academia specifically, but in general, we often think about the complications. Like we're not, the population is not, exponential, and there's demographics and all sorts of things. These are all usually second order effects that are important if you want to be very accurate. And, if you're a professor and you want to do the exact calculation, it's probably right. But usually you just want to get out of the room, give an answer and continue with your life. And for that, usually it's just fine to be simplistic or to use a simplified relation. And for that holds very much. And the fact that, different people live different duration in life, not so important. The fact that the pool size number of people in humanity is not constant, we could see how much effect does this give. Probably not, wouldn't change the order of magnitude, maybe even much, much less, not even a factor of 2. Okay, so in that spirit, and also with that, I send you into the week. So just to be clear, you have a new exercise waiting for you. Just in the big picture, there's this exercise and another exercise, and you're done with exercises. Okay, so just 2 exercises left. So hold on with that. We have 2 more lectures. You should probably already start thinking about ideas for final project, but we have time. Also, if you're unclear and also if you're afraid, you come talk with, Lior or Sam. We have experience with that. It would grow on you. So no need to panic. And we'll send you some examples. You can see how people did it last year. תודה רבה.